Hello, I'm Dr. W. John Martin. I would like to explain my research. It began in 1986 and soon led to the detection of viruses that are not effectively recognized by the immune system. Consequently, the viruses do not evoke inflammation, the hallmark of an infection. The patient's cellular immune system fails to respond, even though the viruses cause cell damage in the brain and other tissues. The viruses are derived from regular viruses, including a monkey virus contaminant from polio vaccines, but have lost or mutated the genes coding for the relatively few components normally targeted by the cellular immune system. I describe this immune evasion mechanism as stealth adaptation. Now you might wonder, if the cellular immune system is not responding, why the patients don't get progressively worse over time. The same issue arose in virus-inoculated animals. They would become sick, but then clinically recover, even though there was no inflammation. So also in virus cultures, the cells would show damage, but then undergo a repair process. Brain biopsies from infected patients showed markedly disrupted mitochondria in the infected cells. Mitochondria are where cells obtain over 90% of the energy from food metabolism. How, therefore, were the infected cells obtaining energy to survive and to suppress the infection? I'd like to summarize or condense some pretty major research. I now know that cells can obtain energy from an alternative cellular energy pathway that is different from the energy obtained from food metabolism. I've also proposed a force in nature that is attracted to separated electrical charges. The fundamental purpose of this force is probably to prevent the fusion and annihilation of electrostatically attracted opposite electrical charges. I refer to this force as KELEA, K-E-L-E-A, referring to kinetic energy limiting electrostatic attraction. Dipolar compounds have separated electrical charges. Certain dipolar compounds can not only attract KELEA, but can transfer energy to nearby fluids, including water. So too can some devices that create fluctuating levels of electrical charges. The absorption of KELEA into water leads to a loosening of the electrostatic hydrogen bonding between water molecules. This adds a dynamic or kinetic quality to the water, which can be converted to chemical energy. If water is sufficiently activated, its now somewhat separated charges can begin to directly absorb KELEA, leading to further activation. It can also activate added water, and this explains the practice in homeopathy of undergoing repeated dilutions. Many individuals have insufficient cellular energy to maintain optimal health. While food provides one type of cellular energy, some functions require energy from the alternative cellular energy pathway. In particular, the ACE pathway provides for non-immunological resistance to infections, including those due to stealth-adapted viruses. Unlike the immune system, the ACE pathway does not cause inflammation that can lead to scarring. Enhancing the ACE pathway is extremely beneficial in preventing scar formation following burns and trauma. When certain tumor cells are provided with additional energy, they will undergo a self-destructive process called apoptosis. This explains the painless regression of tumors being seen in some ACE pathway treated cancer patients. When nerve cells have insufficient energy, they initially become hyperresponsive, followed by hypoactivity and quiescence. Both types of changes can be potentially corrected using the ACE pathway. Indeed, it is important that we proceed with clinical trials using the ACE pathway in patients with brain illnesses, including children with autism. Enhancing the ACE pathway can also potentially alleviate those medical conditions in which the energy from food metabolism is impaired. This can occur if the body has inadequate oxygen, as for example in chronic obstructive pulmonary disease or COPD, or if the blood supply is impaired, as in cardiovascular and cerebrovascular diseases, or if metabolism is generally inefficient, as in diabetes. The practical question is how best to activate the ACE pathway. I personally prefer the use of dipolar compounds, including humic acid, to activate the water. These compounds can be removed before drinking the water, or the water can be inhaled as a fine mist or applied directly to the skin. For more severe acute conditions, the activated solutions can be injected intravenously. Alternatively, the solutions can be mixed with certain dyes, including a dye neutral red, placed into a plastic bag that's laid onto the skin and illuminated with ultraviolet light. An important question is, how does the body naturally support its ACE pathway? And can we assist in this effort? I believe that certain types of fluctuating electrical activity in the brain acts as an antenna to bring energy into the body. Laughing yoga seems to do this. The body can also make KELEA attracting mineral containing materials that self-assemble into particles, fibers and threads. I refer to them as ACE pigments, and they can be detected on the basis of ultraviolet fluorescence, especially with neutral red dye. The message from this talk goes beyond the dictum that man does not live by bread alone. Rather, it outlines a new paradigm in medicine, one that is based upon the ACE pathway. We can all benefit by learning more about this pathway. Some information is available in a book entitled Stealth Adapted Viruses, Alternative Cellular Energy, and KELEA Activated Water. Thank you for listening.